tools of Neanderthals were quite similar in the beginning of their history and in the end of their history, over 400,000 years. And they were also very similar over their entire distribution from Western Europe to Central Asia. The materials differed, but what they made were quite similar. And that then changed with modern humans. With modern humans, technologies start changing rapidly. And we see that also become regionalized. Specialists can immediately see if a modern human stone tool would come from Southern Europe or from Central Asia. Art is another thing. There are some art that is probably done by Neanderthals. I sometimes joke and say that looks pretty much like modern art because I can't see what it depicts. But figurative art comes only with modern humans, where we immediately see what is depicted. And of course, modern humans become very numerous and spread to the entire habitable part of the planet. All earlier forms of humans never, perhaps with one exception, but never crossed open water where you don't see land on the other side. They never came to Australia. They never came to Madagascar, never to the Americas. Modern humans within 40,000 years and spread to everywhere, out in the Pacific to the last little island in the Pacific. There is something quite special with modern human behavior or sociality or something. And a dream is then to understand some little aspect of that, perhaps, by studying these changes that happened here. So if we just list everything that changed here, and changed so drastically, so everyone today carry it. So we're looking for genetic changes that everyone today, as far as we know, on the planet carries, but Neanderthals didn't have. Then that list is not that long. It's around 30,000 nucleotide changes. So you can look through them in the computer in an afternoon. Your problem is you don't understand what you look at all, because we're very bad at inferring function from just a change in the genome. But we want to try this anyway. And I just want to illustrate that with one group of changes that are particularly likely, perhaps, to have consequences, and that is changes that change amino acids in proteins. So they change the structure of the molecules that do much of the functions in the body. And there are just approximately 100 such changes. So you can list them all in 87 genes here. And when you look at that, one thing that struck us was that this is a rather short list. There are like 20,000 proteins at least encoded in the genome. These are just 87 of them. And 3 of them here has to do with cell division, how chromosomes separate when 2 cells divide. And not only that, the only protein here with 3 amino acid changes is here. One of only 5, I think, with 2 changes, and one has 1. So there are 6 of these approximately 100 amino acid changes that affect proteins that have to do with chromosome segregation, which was totally surprising to me. I thought if something has changed recently in human evolution, it's not going to be something as basic as segregation of chromosomes. But of course, we're particularly interested then about any effects that one might have in brain function or brain development, because we suspect, after all, that it's something with modern human cognition or sociality that is special. So if we then think about how neurons sort of are formed when the brain develops, there are stem cells that divide in the sort of apical parts of this epithelium and then migrate up and eventually form mature neurons here. And going back 5-6 years, we've been very interested in studying this process in collaboration with a group at the Max Planck Institute in Dresden, Wieland Huttner's group, and comparing that to apes. But of course, you cannot study the developing brain of apes at all, and in humans, very, very difficult to do. So we have made use of an invention from Japan here, induced pluripotent stem cells invented by Professor Yamanaka in Kyoto, where you can take cells from, for example, an ape, make them into stem cells that you can then differentiate to different tissues, for example, into cerebral organoids, which are these little balls that are like early stages in brain formation that you can study. So you can then look in this epithelium here and see these apical parts here where the stem cells divide and then they migrate up and continue dividing and eventually form neurons. And we at the time then compared chimpanzees and orangutan organoids to human organoids. And as you would expect, they are almost identical. It's almost no differences you can see. But Fernando Bermudez, who studied this in Wieland's group, together with us, found something interesting. And I should see if I can now start this. Yes, so we can go back here. So you can now, what you look at here are the chromosomes, stained chromosomes in these stem cells when the chromosomes segregate. And when I play this video, you will see the chromosomes line up and then be pulled apart at the end of what we call metaphase into the two daughter cells. So here are the chromosomes, they condense, they form this metaphase plate here, and there they are pulled apart. And if you follow these two sides here, this is a chimpanzee and the human, you would have seen that this metaphase time here was shorter than here. So there is this, we found back in 2016 that the time when the chromosomes line up is longer in humans than in chimpanzees. It is as if they pair up more carefully and take more care before they're pulled apart. So we were then very excited to see that when we compared to the Neanderthals, there were these 3 genes involved in this process that had amino acid changes. So I then want to share with you for the first time actually talking about some unpublished results that we are about to hopefully publish. So what we have done is then create a mouse, or different mouse lines actually. The mouse, of course, looks like the chimpanzee at these positions, have the ancestral amino acid positions. So in one protein, we then changed the mouse to the modern human version here. The other protein, there are 2 places that we changed to the modern human, and in a third protein, there is 3 positions. So we then created mice that carried each of these chromosomes separately or combined them by crossing the mice to each other so they had 2 or all 3 together. And then looked again at the segregation of chromosomes as I just showed you. And what we then find is that the mouse that have all these 3 proteins, humanized form, get a longer metaphase, exactly as we would expect. The human have a longer metaphase than the chimpanzee and the mouse. When we actually have only 2 of these proteins humanized, it is also just as long. And when we do just these single cases, it's not as long. One seems to have no effect, the other one perhaps a partial effect here. So it then seems that these changes in these 2 proteins are crucial here. So we of course then wanted to see this also in the human genetic background. So we then changed in the stem cells these positions, 2 in this protein and 1 in this position, and made organoids and looked at chromosomal segregation in these apical progenitors. And indeed, what you find is that when you change them back to the Neanderthal chimpanzee-like state, your metaphase gets shorter and other parts of the cell division is not affected. And you may then ask, does this have consequences for the segregation of the chromosomes? And it seems to have, because you can then look later on in cell division when you see a chromosome that is not properly segregating but get lost between the cells here, or fragments, large fragments of chromosomes, and count those. And if you then look in the ancestralized organoids, you see more of these. It's a very small number, but you see a tendency for having more such missegregations in the Neanderthal-like brain stem cells than in the normal modern human controls. So it then seems, if we look in these apical progenitors here, that if you compare humans to Neanderthals, you would have a more accurate segregation of chromosomes in the modern human state than in the Neanderthal state. And this seems, by the way, to be specific for these cells. If we look in blood cells, for example, we don't see this, or undifferentiated stem cells, we don't see this. So it's, of course, very tempting to speculate that this may have something to do with how the brain later functions. That it may be that these cells were missegregating chromosomes, these neurons simply die and are lost, for example, or functions poorly in the later development of the brain. But that's exactly what we are since a few months with this. So we're sort of struggling with then looking at the downstream consequences of this. But this is sort of an illustration of what we are then working with. And I hope I have sort of convinced you then that having the genomes of our closest evolutionary relatives, Denisovans and Neanderthals, is interesting because it allows us to study things that are unique to modern humans, like the last example, as well as things that changed on the lineage to these extinct forms of humans and then came over to present-day humans, like the COVID example or the progesterone receptor example. And as you've seen, the way to study this is then to study variation in present-day humans, to see, for example, what the Neanderthal variant has for consequences. But then for things that are in present, changed in the human line, and is present in everybody today, you reduce to then study, for example, changes in stem cells that differentiate or in humanizing mice. And